Dr. Mike Hildebrand was preparing for a career teaching high school science until a fourth-year undergraduate research project with Tony Stay in the biology department opened up the world of research to him. In this talk, Mike will highlight his UFE honors thesis work, investigating how pyrethroid pesticides block channels that regulate excitability in the brain, and briefly explain how this led towards a career investigating mechanisms and new treatments for pain. Dr. Hildebrand.
Thank you so much, Gary, and the organizers for allowing me to speak here today. I'm between you and awards, so I'll try to keep it brief. And I also want to echo Gary's words. I'm just inspired by hearing all of you speak today. Just the depth and breadth of your research that you've presented, the quality of those presentations. This is a tough act to follow, all of you. And the posters that we saw earlier today, like really well done. And so I'm going to just share a little bit about my research journey. If I can see my slides, I'm going to move a little bit so I can see my slides and sheet a little bit. About my research journey and less on the science, more on a couple lessons learned along the way, because maybe that can be an encouragement to each of you as you're kind of navigating these. And so I'll talk about how I moved from, as Gary was saying, my first exposure to research as an undergrad here at UFE. This building didn't exist, so over in a different building at UFE, looking at the effects of pesticides on the nervous system, and ultimately how that led to now the research that my lab is doing now, looking at spinal mechanisms of pain to try to identify new treatments for pain. So when I first came to UFE, like some of you, I had a clear career goal. I was going to teach high school science. And maybe like many of you now, by the time I was near the end, I was more confused than had answers. A lot of wrenches were thrown in there. Some doors were closed too, and I can talk about that on the side after if any of you want to hear those stories. But part of that wrench in the plan was this exposure to the world of research. And so there's me a couple decades ago rocking the sideburns and that nice fleece sweater there at a research day like this over 20 years ago. And so I did an honors thesis, or not an honors thesis, a directed study, an undergraduate directed study under the supervision of Tony Stay in biology. And I'm sure, as similar to many of you, when I started, I felt like I was thrown into the deep end. I remember just that first day getting a solution and being like, well, where's the manual? Like, it doesn't say how much to add. Like, it's just concentrations, and kind of feeling like, what are we doing here, right? And trying to learn as we go. And so my project, I won't go into too much details, but it involved actually taking eggs out of xenopus frogs and injecting DNA into those eggs as a factory to make certain channels, and then to look at the effects of pesticides on those channels. And so kind of in the process of that, what I discovered was this world of research, how challenging it was, and frustrating at times, right? But how motivating it was, the thrill of discovery, having that kind of one step forward, two steps back. But I really got hooked. And so I was a first generation university student and opened up this world that I had never even considered type thing. So that was one of the things, just this exposure to research and the starting of how it might be part of my future plans. And the other big thing from that journey was just the power of mentors, both in science, but also in life. And I think UFE, that's what makes this place so special. And that's why I love coming back here every time, is because we have professors, we have staff who are all about investing in students. Sorry. It's close to my heart just because of how much it impacted my life. I did not anticipate getting emotional. So I'll take a collective breath here. But and so what that meant was Tony providing input in the research side, but also, and so this is the encouragement to you that some of you are leaving UFE, right? You're finishing your research, but that doesn't mean those mentorship relationships need to end. They can be those sounding boards you turn to at various stages in your life. And actually, Tony and I have stayed connected. He was over at my house in Ottawa. I'm going to dinner tonight with him and Michelle. And so these are relationships that can continue. I gotta keep an eye on time so I don't go too long. So I'll give you a little snapshot of what the science involved in this honors thesis project. We are really looking at a specific type of pesticide found in many types of household pesticides, like those mosquito coils that I was showing, called elethrin. It's a pyrethroid pesticide, and elethrin is known to act on a type of channel in your nervous system, voltage-gated sodium channels. And so our question was, does it also block these other type of channels, voltage-gated calcium channels, which are critical for mechanisms of learning, plasticity, excitability throughout your nervous system? And so we initially put those channels into frog eggs and tested the effects of pesticides, like some of the themes that emerged here. It worked a few times, it didn't work several times. So I actually wrapped up this study. I can't see. Yeah, I wrapped up this study looking at the effects when I had started my PhD. And so we expressed the channels in cells, in cell culture, in cell lines, like you've also heard today, and we showed that these pesticides potently at the micromolar level block the different types of channels. I can't really point, but there's basically 3 different flavors of channels there, and they potently block all the types of channels of these different calcium channels. T-type channels, a PQ type that are found in synapses, and L-type that are found in the heart and in other places. And that block was reversible as well. And we also looked at how it blocks the channel, but I won't go into the details there. But kind of, once again, kind of bringing it back to this, that led to my first presentation, which was similar to a day like today, but a lot smaller, I would say, because that was a couple decades ago. It's amazing to see the growth. So that was my first presentation, research presentation, and several that followed on that story, and it was also my first paper, and I'm proud to say that paper is one of actually my more highly cited papers because of the implications it has for understanding how these pesticides can work in insects, but also potentially toxicity. So people in the toxicity field have kind of used that as a starting point for other research. So then following that time, I went on to kind of actually through a connection that Tony had, but as I got exposed to thinking about graduate school, because I had no world of the idea of going to graduate school. And so I went on to do a PhD with Terry Snutch at UBC, looking at one of these types of calcium channels, T-type calcium channels, and the role of signaling in the brain. And so I wrapped up that study you just saw, my honors thesis study. Then I had this project where I was looking at a new blocker for a new type of channel, and I was doing all of these experiments. And we were seeing effects, but the concentration, you increased the concentration and it didn't really increase the effects, and we were trying to figure out, and I realized, so this is a good lesson of why controls are so important in science, because I'd done a control, but not for that channel I was recording from. And so I took away the drug and tested the solvent, and it had the exact same effect. And so I had a year and a half of science kind of out the window. And so this is where your mentors are key, because I can remember clear as day calling Tony and saying, like, I'm going to drop out. Like, I have nothing to show for it. It's a year and a half in. And he said, Mike, science isn't linear. And I would say life isn't linear, right? Especially our career paths. We think it's one to the next, but it really isn't. And so those words rang true, and I find myself, I quote that for my students now when they hit those roadblocks, and when you think there's no way I can finish, what am I going to do? And it was so true. I finished half of my PhD thesis in 6 months, right near the end, because you're developing all those skills that you're learning, all those experiences that you're learning. You'll develop them, so then near the end you hit something, and now you're equipped to really see it and to go through and avoid all those pitfalls that you can only learn by going through them. So then just a couple things about my PhD. Near the end I wrote a review, and so this is just an example. I wasn't studying pain, but my boss was like, do you want to write a review on the channel you're studying in pain? I said, okay. So I wrote a review paper on that, and that opened up the world of pain research to me, which I hadn't considered. I also, near the end of my PhD, Tony was on sabbatical, and so he needed someone to teach his anatomy and physiology course. Said, Mike, would you be interested? And I said, okay, I'll give that a try. And because I was interested in teaching, that was my original goal, teaching in high school. So I took that on, and man, was it a lot of work. And it involved dissecting. It had a lab with mud puppies and sharks and dogfish. And so it was a ton of work, but I learned a lot, and I actually got exposed to teaching and got the real bug there of, once again, doing really helps you figure out kind of what you're good at, and it also helps figure out what you're passionate about and your unique niche. So I'd say those things that can seem like detours. You never know how they're going to come together to create unique opportunities. And so I continued on then after my PhD. I wanted to stay in Vancouver, so I did an industrial postdoc with my PhD supervisor. He also had a company looking at blocking these channels for pain, Terry Snutch, which is shown there, a company called Neuromed. So I did an industrial postdoc. That's when at that stage I kind of thought, okay, maybe I'll take the risk and go that academic route. And so at that time we had 2 boys, and we uprooted from Vancouver and we went to Toronto to Sick Kids. That's Mike Salter. So those are another 2 of my mentors. And so that was actually in the Philippines a year ago, where it was really special to have them both together at the same conference and get to thank them too. And so basically the story from this part of my career is just finding that unique niche. It was studying these different types of channels. Now I kind of had different channels, a different assay of spinal cord excitability, and realizing very little was known about how those channels function and how they could potentially be targets for pain that we could potentially treat. And so that's what led to when finally that dream job came up at Carleton University, where it could combine my passion and past experiences in research and teaching and my exposure to really good mentoring to tie it all together to now have my own research team to unpack this. And I think I'm close to my time, so I'm not going to talk about my pain, but we can talk about that separately, and I'm also going to be highlighting this on Saturday. But basically, I'm going to skip through the actual pain research that I'm doing now, because you can see there was a lot of slides. I was very ambitious. But I'll just say, the take-home message of what we're doing is we're using human tissue from organ donors to bridge that gap between preclinical experiments in rats to, are these donors and these potential targets conserved in human tissue? And we're finding sometimes they're conserved across species, sometimes they're not. Sex is also biological sex is a key variable, and we're seeing some differences across sex as well. And that's an example of a story where there's a difference across sex. But I wanted to get to this last slide because this is the people that make the science happen. It's not me, it's the team, just like it was every one of you making the research happen in your teams. And so they are the ones who drive it forward, along with obviously collaborators and funding that make it all happen. So thank you so much for being here today, for letting me be here today.